Amino acids serve as building blocks for proteins. Amino acids are precursors for many nitrogen-containing compounds that have important physiologic functions. They include porphyrins, neurotransmitters, hormones, purines, and pyrimidines. Today, we will cover two topics. First, we will look at porphyrin metabolism. Porphyrins are cyclic compounds that bind metal ions. Most prevalent porphyrin is heme. Porphyrins are prosthetic groups for hemoglobin, myoglobin, cytochromes, and catalase. Porphyrins are cyclic structures formed by joining four pyrrole rings through methylene bridges. Each of these is a pyrrole ring, and this is methylene bridge. Different porphyrins have different side chains attached to the pyrrole ring. For example, a uroporphyrin has acetate and propionate side chains. Here's acetate and here's propionate side chains. A represent acetate and P represent propionate. There are four ways side chains can be distributed around the pyrrole rings, named with Roman numerals 1 to 4. Here are uroporphyrins 1 and 3. What's the difference between 1 and 3? The A and P are reversed in the D ring in uroporphyrin 3 compared to uroporphyrin 1. It turns out type 3 porphyrins are physiologically important in humans. Type 1 may build up in some diseases. Structural features of porphyrins are summarized in this slide. Again, only type 3 porphyrins are physiologically important. Porphyrinogens are the reduced precursor of porphyrins. They are colorless and unstable. They are readily and irreversibly oxidized to porphyrins that are red and stable. Only one porphyrin, protoporphyrin 9, is retained and is converted to heme. Others are excreted in urine or bile. Heme is the final product of porphyrin biosynthesis. Now, let's move to part 1a. We will look at heme biosynthesis. The rate-limiting step for biosynthesis of heme is the formation of delta aminolevulinic acid from glycine and succinyl-CoA. 4 steps take place in mitochondria. Heme biosynthesis occurs in the liver and erythropoietic cells of bone marrow. This is an overview of heme biosynthesis. The first step and the last 3 steps take place in the mitochondria. The rest 4 steps occur in the cytoplasm. Here are enzymes involved in heme biosynthesis. ALA synthase is the rate-limiting enzyme. It catalyzes the first reaction. Succinyl-CoA and glycine condensed to form delta aminolevulinic acid with removal of 1 carbon dioxide. This involves a decarboxylation step and requires pyridoxal phosphate as a coenzyme. Here is integration with phospholipids. Decarboxylation of phosphatidylserine also requires pyridoxal phosphate. 2 drugs interfere with pyridoxal phosphate, penicillamine and isoniazid. Here is integration with microbiology. Isoniazid is a drug to treat tuberculosis infection. Because isoniazid interferes with pyridoxal phosphate, patients treated with isoniazid should receive vitamin B6 as a supplement. ALA synthase is inhibited by heme and heme. Heme is a derivative of heme. Drugs metabolized by cytochrome P450 monooxygenase system, such as phenytoin, phenobarbital, and rifampin, increase synthesis of ALA synthase. This is due to the effect that drugs bind xenobiotic sensing nuclear receptors. These nuclear receptors increase transcription of ALA synthase gene. In the second step, two molecules of ALA are condensed to form porphobilinogen. Porphobilinogen is a common precursor of all natural tetrapyrols, including hemes, chlorophylls, and vitamin B12. This reaction is catalyzed by ALA dehydratase. This enzyme is inhibited by heavy metals, especially lead. In the third step, four molecules of porphobilinogen are condensed to form linear hydroxymethylbilane while releasing four molecules of ammonia. This reaction is catalyzed by porphobilinogen deaminase. In the fourth step, hydroxymethylbilane can be spontaneously converted to uroporphyrin 1. This compound can also be converted to uroporphyrin 3, catalyzed by uroporphyrin 3 synthase. This reaction is much faster than non-enzymatic reaction. In the fifth step, carboxyl groups from 4 acetate side chains are removed by uroporphyrin 3 decarboxylase to form coproporphyrin 3. This enzyme also catalyzes conversion of uroporphyrin 1 to coproporphyrin 1. Uroporphyrin decarboxylase is an unusual decarboxylase since it does not require any coenzyme. Deficiency in this enzyme causes porphyria cutaneous tartar. In the sixth step, carboxyl groups from 2 propionate side chains are removed. 2 single bonds are converted to 2 double bonds. This reaction is catalyzed by coproporphyrin 3 oxidase. This reaction occurs inside mitochondria. In the seventh step, hydrogens are removed from B, C, and D rings of protoporphyrin 9. This gives rise to protoporphyrin 9. This reaction is catalyzed by protoporphyrin 9 oxidase. In solution, protoporphyrin 9 is colorless, while protoporphyrin 9 is red. In the eighth and final step, ferrous iron is inserted into protoporphyrin 9 to form heme. This reaction is catalyzed by ferrochelatase. This enzyme requires ferrous iron as well as reducing agents, including ascorbic acids and cysteine residues. This enzyme is inhibited by heavy metals, especially lead. Here is a summary of heme synthesis. The first two steps lead to formation of porphobilinogen. In the next two steps, 4 molecules of porphobilinogen are converted to uroporphobilinogen 3 with formation of the first tetrapyral ring. In the next several steps, uroporphobilinogen 3 is converted to protoporphyrin 9. Finally, ferrous iron is inserted into protoporphyrin 9 to form heme. In this whole process, several byproducts are produced, including uroporphyrin 1, coproporphyrin 1, uroporphyrin 3, and coproporphyrin 3. Buildup of these byproducts in patients can cause photosensitivity. Here is a clinical correlation. Porphyrias are inherited or acquired deficiency in heme. They are classified as hepatic or erythropoietic. All are autosomal dominant except congenital erythropoietic porphyria. Porphyrias can be divided into two groups. First group does not have photosensitivity. For example, ALA dehydratase deficiency caused by lead poisoning and acute intermittent porphyria caused by porphobilinogen deaminase deficiency do not have photosensitivity. Conditions in the second group all have photosensitivity because of buildup of various porphyrin byproducts. The first group of porphyrias is represented by acute intermittent porphyria. It is caused by deficiency in porphobilinogen deaminase. Enzyme deficiency leads to accumulation of ALA and porphobilinogen. Patients with this condition have abdominal pain and neuropsychiatric disturbance, but without photosensitivity. This condition is often precipitated by barbiturates and ethanol. Second group of porphyrias has accumulation of tetrapyral rings. These include congenital erythropoietic porphyria, porphyria cutaneous tarda, and hereditary coproporphyria. Porphyrias in this group share common features. They include photosensitivity, an increased ALA synthase activity, and accumulation of upstream intermediates. This is true for acquired porphyrias, such as lead poisoning, as well as hereditary porphyrias. Treatment of porphyrias include intravenous injection of heme to decrease synthesis of ALA synthase, use of beta-carotene as an antioxidant. Patients with porphyrias should avoid sunlight. Now, let's move to part 1b. We will look at degradation of heme. Most of heme is derived from hemoglobin. Breakdown of heme to bilirubin occurs in macrophages. Unconjugated bilirubin is carried by albumin and transported to the liver. Bilirubin is taken up by the liver and conjugated with glucuronic acid. Conjugated bilirubin is actively secreted into the bile and then to the intestine. In the intestine, glucuronic acid is removed by bacteria. The resulting bilirubin is converted to urobilinogen. Some of the urobilinogen is reabsorbed by the intestine and enters the portal blood. A portion of this urobilinogen participates in the enterohepatic urobilinogen cycle. The remainder of the urobilinogen is transported by the blood to the kidney, where it is converted to yellow urobilin and excreted, giving urine the yellow color. Urobilinogen is oxidized by intestinal bacteria to brown steatobilin, giving the feces a characteristic color. Here, we will take a closer look at this process. Heme is degraded in two steps to bilirubin by macrophages. Bilirubin is highly lipid soluble and carried to the liver by albumin. In the liver, bilirubin is conjugated with glucuronic acid and excreted into the bile. There are three carrier proteins. One is haptoglobin that carries alpha-beta dimer of hemoglobin. Hemopexin carries free heme. Albumin carries free bilirubin. Here I need to mention haptoglobin. In the plasma, hemoglobin dissociates into alpha-beta dimers, which can escape through the glomerular filtration system of the kidney to appear in the urine. To prevent this, haptoglobin binds the dimer and delivers to the macrophages for degradation. The first reaction is cleavage of heme ring between A and B pyrorines. This reaction is catalyzed by heme oxygenase. It is located in the ER. This reaction requires NADPH. The products are biliverdin, carbon monoxide, ferrous iron, and NADP+. This is the only endogenous source of carbon monoxide. Second reaction reduces the central methylene bridge of biliverdin, giving rise to bilirubin. This reaction also requires NADPH. In the liver, bilirubin is conjugated with two molecules of glucuronic acid, donated from UDP glucuronic acid. This reaction occurs in two steps. It is catalyzed by UDP glucuronosyl transferase, or UGT. Plasma bilirubin levels can be determined by using the Vandenberg reagent, diazonium salt. This chemical can react with bilirubin to form a colored complex. Only conjugated bilirubin is water-soluble and reacts directly with this reagent. This will give direct bilirubin. To measure the unconjugated bilirubin bound to albumin, alcohol is added to release bilirubin into solution, where it can now react with the Vandenberg reagent. This will give total bilirubin. Indirect bilirubin can be determined by subtracting direct bilirubin from total bilirubin. Here is clinical correlation. Hyperbilirubinemia causes jaundice. It is yellow discoloration of the eyes and skin, as shown here. Direct and indirect bilirubin values are used in the differential diagnosis of hyperbilirubinemia. Normal range of direct bilirubin is below 0.3 mg per deciliter. Hyperbilirubinemia can be caused by either increased bilirubin production, decreased uptake bilirubin into the liver cells, impaired conjugation, or interference with secretion of conjugated bilirubin. Conditions causing jaundice can be divided into three groups: prehepatic, hepatic, and posthepatic. Prehepatic conditions are those that increase hemolysis. Hepatic conditions include those have reduced uptake of bilirubin into liver, such as Rotor syndrome, decreased conjugation of bilirubin, such as Gilbert syndrome and Crigler-Najjar syndrome, and decreased secretion of bilirubin in the bile, such as Dubin-Johnson syndrome. Posthepatic conditions are associated with biliary obstruction. Prehepatic conditions causing jaundice also cause increased bilirubin in bile, increased urobilinogen, increased urobilin in the urine, and unconjugated bilirubin in the blood. Jaundice in this category is most often caused by hemolysis. Rotor syndrome is caused by mutation in bilirubin transporters, specifically organic aniline transporting polypeptide 1b1 and organic aniline transporting polypeptide 1b3. Without these proteins, bilirubin is not efficiently taken up by the liver. Conditions with impaired conjugation are most often caused by defects in UGT. Patients with Gilbert syndrome have increased unconjugated bilirubin levels. It is caused by a reduction in the UGT activity. Those mutations are hypomorphic. Crigler-Najjar syndrome is a condition in which conjugation is impaired. There are two types, type 1 and type 2. Type 1 is most severe. It is caused by a loss of function mutation in the UGT1A1 gene. In this condition, unconjugated bilirubin level can reach about 20 mg per deciliter. Preemies, premature born infants, especially have low UGT activity for the first 2 weeks of life. Bilirubin can diffuse into basal ganglia and causes encephalopathy. Treatment includes blue fluorescent light, which converts bilirubin into more polar compound that can be excreted in bile without the need for conjugation. Dubin-Johnson syndrome results from inability of hepatocyte to secrete bilirubin. It is caused by a mutation in the canalicular multidrug resistant protein 2 or multispecific organic aniline transporter 1 protein. Patients with Dubin-Johnson syndrome have black-brown liver cell pigmentation. Biliary obstruction causes backup and reabsorption of conjugated bilirubin. As a result, blood level of conjugated bilirubin increase. Now, let's move to part 2. We will look at synthesis and degradation of other amino acid derivatives. This is creatine. It is derived from arginine and glycine in 2 steps. In the first step, guanidino group is transferred from arginine to glycine to form guanidino acetate. In the second step, the nitrogen is methylated using SAM as the methyl donor. This gives rise to creatine. Creatine can be phosphorylated by creatine kinase to form creatine phosphate. Creatine phosphate is a high-energy compound, primarily in muscle and brain. Excretion of creatine is in proportional to muscle mass. Histamine is a powerful vasodilator, paracrine factor, and neurotransmitter. This is histamine. It is derived from histidine through decarboxylation. This reaction is catalyzed by aromatic L-amino acid decarboxylase, or AADC. AADC also catalyzes several other decarboxylation reactions, such as L-tryptophan to tryptamine, L-dopa to dopamine. AADC requires pyridoxal phosphate as a coenzyme. Serotonin is a neurotransmitter, vasoconstrictor, and agonist of platelets during coagulation. This is serotonin. Serotonin regulates mood, appetite, and sleep, as well as cognition. It also plays a role in control of blood pressure and coagulation. Serotonin is derived from enterochromaffin cells in GI tract and stored in platelets. A small amount of serotonin is also produced by the central nervous system. Serotonin is derived from tryptophan in two steps. In the first step, hydroxyl group is added to the tryptophan, giving rise to 5-hydroxytryptophan. This step is catalyzed by a hydroxylase. This enzyme requires tetrahydrobiopterin as a coenzyme. Second step is decarboxylation. It is catalyzed by AADC that I described to you earlier. AADC requires PRP as a coenzyme. Carnitine is required for transport of long-chain fatty acids into mitochondria. It has antioxidant properties. Carnitine is made from a lysine derivative, N-trimethyl-lysine, in 4 steps. The first step is addition of hydroxyl group, catalyzed by hydroxylase. Second step is cleavage between alpha and beta carbon, catalyzed by an aldolase. Third step is oxidation of the aldehyde group to form an acid. This step is catalyzed by a dehydrogenase. Final step is addition of a hydroxyl group, catalyzed by dioxygenase. Catecholamines include dopamine, epinephrine, and norepinephrine. Dopamine and norepinephrine are neurotransmitters in the brain and autonomic nervous system. Epinephrine is a hormone. Epinephrine and norepinephrine are synthesized by adrenal medulla in response to fright, cold, and low level of glucose. Catecholamines are derived from tyrosine in 4 steps. The first step is addition of a hydroxyl group. It is catalyzed by tyrosine hydroxylase. This enzyme requires tetrahydrobiopterin as a coenzyme. Second step is decarboxylation, catalyzed by AADC that I described to you earlier. Third step is addition of a hydroxyl group. It is catalyzed by dopamine beta hydroxylase. This enzyme requires vitamin C as a coenzyme. The final step is transfer a methyl group to norepinephrine to give rise to epinephrine. This reaction is catalyzed by phenylethylamine N-methyltransferase, using the SAM as their methyl donor. Catecholamines can be inactivated by oxidative deamination, catalyzed by monoamine oxidase, or MAO, with release of ammonia. As shown here, catecholamines can also be inactivated by O-methylation, catalyzed by catechol O-methyltransferase or COMT. The metabolic products of these reactions are excreted in the urine. MAO inhibitors act as antidepressants. MAO inhibitors inactivate MAO, permitting neurotransmitter molecules to accumulate within a presynaptic neuron, thereby increasing their availability. Melanin is a group of natural pigments in the skin, hair, and eye. These pigments protect cells from harmful effects of sunlight. There are two common forms of melanin, eumelanin and pheomelanin. Melanin is derived from tyrosine. It is synthesized by melanocytes. Synthesis of melanin takes place in two major stages. First stage converts L-tyrosine to L-dopa-quinone and involves two steps. In the first step, tyrosine is converted to dopa. In the second step, dopa is oxidized to dopa-quinone. These two steps are catalyzed by tyrosinase. Second stage leads to formation of the pigment. It involves several smaller steps. Here is clinical correlation. Mutations in tyrosinase cause albinism.